Hello everybody, so today we'll be doing enzymes. So hopefully we remember what enzymes are. Okay, let's do a quick overview. Enzymes, okay, so remember enzymes are proteins. So now that we got that, you remember what type of biomolecule that is. Let's get into what an enzyme is. So enzymes do a million different things, so they're super important for our bodies, okay? So first off, an enzyme, let's look at this picture right here. Okay, so an enzyme is right here, and there's always going to be an active site. Okay, so this part right here that this arrow is pointing at, this is an active site right here. Okay, and the thing that attaches to an enzyme is called a substrate. Okay, so you have an enzyme, you have a and substrate, and there's an active site. Okay, so substrate is what the enzyme attaches to. Okay, and the active site is where the enzyme, substrate bind together. Okay, so remember, okay, an enzyme is a protein, okay, and it's going to, what it's going to do is break down substrates, and these substrates are sugars or simple sugars. Okay, so make sure you understand that. So, for instance, you may have lactose, and it's going to break down the simple sugar called you may have lactase, and it's going to break down the simple sugar called lactose. Okay, so make sure you understand that substrate is actually a simple sugar. Okay, awesome. Okay, so this is how the actual enzyme binds together. So I'm going to circle this because this is really important to know. Okay, so what happens is you have your substrate, and it's going to find the enzyme. Okay, so they're going to be specific. So the substrate has to bind to a specific enzyme and vice versa. So specific substrate. Okay, so it has to bind to a specific substrate, it being the enzyme. Okay, so what happens is over time you have the enzyme, the substrate are going to bind together, and then they're going to break apart, creating products. So your substrate's going to bind, going to go through the reaction, and it's going to break apart. Notice the enzyme does not break, the products break. Okay, awesome. All right, so here we have what are enzymes and how do they work. Okay, so enzymes are catalysts, so make sure you highlight this word catalyst because you can see this as well. So enzymes or catalysts, okay? So these are proteins made out of amino acids, okay? So they help speed up reactions. So let's highlight that. They help speed up reactions. Let's underline it too. By lowering the activation energy. Okay. So we are going to highlight this part that says lowering. So they're going to speed up the reaction by lowering the activation energy. Okay, and activation energy is what we call start energy. Okay, all right, so right here we have the enzyme without an enzyme. Okay, so we have a reaction without an enzyme. So let's put a little thing here. So let's say no enzyme. Okay, and down here we have one with an enzyme because if you notice, it has sped up reaction. So down here we have one with an enzyme. Let's use a different color. So here we have with enzyme. Okay, so in between here, we have what is called the activation energy. So right here, this little section is called the activation energy. Okay, so we have the activation energy, which is helping it to speed up the reaction. So as you can see, you speed it up because it's going to finish first, and you lower the activation energy. Okay, so it finishes first, and you lower the activation energy. Okay, so the bottom one, this bottom one right here, is the one with the enzyme. Okay, because it speeds up and lowers the activation energy. Okay, so it says are reusable. Okay, so enzymes never change. So enzymes are never broken down, okay? So they're reusable because they're not going to be broken down during the reaction. They're going to stay the same no matter what, okay? And they are specific, like I showed you earlier. They must bind to a specific substrate. Okay, so they must bind to a specific substrate. They are substrate specific. Okay, so let's look at here one more time. So the substrate up here, that substrate up here has to be specific. So that's why I told you, if you have a lactase, it would have to bind with a lactose, okay? Okay, so we have an example down here. So catalase break down hydrogen peroxide, okay? Waste products into cells. So this is an example. So for instance, once again, we have lactase. This would break down lactose, okay? And protease breaks down proteins. Okay, so remember, a substrate is a simple sugar in this case. So the enzyme is going to break down a simple sugar. Okay, good. So enzymes, how do they work? This section right here will be very important, okay? So you need to use this for your test, okay? So factors affecting enzyme action, okay? So one of the main things that are going to cause it, we'll talk about that in a second, sorry. Some factors that can cause enzymes to be denatured. Ooh, big word. Big word. We have this word denatured. Let's figure out what that means. Okay, so denatured, I'm going to rewrite it. You need to rewrite it as well. Denature means the enzymes stop working. Okay, so denature means the enzymes stop working. So that's important. So let's figure out how they stop working. So we have temperature. Okay, so enzymes work best between certain temperatures. Okay, so 37 degrees Celsius, by the way, is just 96 degrees Fahrenheit. Okay, so it's best in humans. So let's say if it gets above this, so let's say it gets hotter than that, temperature can affect it and denature it. We have pH levels. Okay, enzymes work at a pH at 7, or some do, especially like amylase, your enzymes in your mouth, work at 7, and pepsin in your stomach work at pH of 2 to 3, because your stomach is acidic, so its pH needs to be lower. Okay, so the third one would be your substrate. Let me rewrite that. Your substrate enzyme concentration. So that means it depends on how much of the substrate and enzyme is available. And the last one is coenzymes. Okay? Helpers such as vitamins and minerals. Awesome. So there are two models that you could see potentially. We are not going to say induce fit. We're not going to say that. There's no reason to. You don't need to know that. That's for next year. Okay, so lock and key model. Once again, this is a substrate must match up perfectly, okay, with the enzyme. Okay, so right here we have this right here is our lock and key model. Okay, so our substrate is going to bind with our enzyme. Remember, our substrate should be a simple sugar like lactose, and it's going to be binding with a enzyme like lactase. Okay, they're going to bind together, and when they're done, their products are going to be different. So remember, the substrate is what breaks into products. The enzyme does not. Okay, so I'm just going to make that note. Substrate breaks into products. Enzyme stays the same. Okay, super important. And there is a second thing here. This is not a model, but this could happen. Okay, so I just need to talk about this really briefly. This is number 2, we'll say, is an inhibitor. Oh, I can't spell. Okay, so what this does, stops the substrate from binding to the enzyme. So I'm just going to draw this just briefly. By the way, this is kind of by itself over here. Okay, so an inhibitor is going to stop the substrate. So for instance, let's say here's our enzyme. Here's our little substrate. Okay, there's going to be something here blocking it. So I want to color this in a different color. Okay, so we have this right here. This right here is your inhibitor. So it stops it from going in. Okay. Substrate enzyme. Okay, so an inhibitor is going to stop the substrate from binding to the enzyme. Okay, so this can be good or bad depending on what's happening in your body. Okay, so once again, these are the notes over enzymes. Okay, whatever I wrote is what you need to write. It's super important. Okay, I have one more. Let's go over it one more time. So you need to know enzymes are proteins. Okay, a substrate binds to an enzyme. Okay, it has to be a specific substrate. It can't be any. It has to be specific. Okay. So for instance, if it's a lactase, it needs to bind with a lactose, and they're simple sugars. Okay, when they bind together and the reaction occurs, they're going to break apart. The substrate breaks apart. It's going to break apart into different little products. Okay, the enzyme stays the same. Enzyme stays the same. Okay, an enzyme or catalyst, so make sure you know the word catalyst. They're just proteins made of an amino acid. What they do is they speed up reaction like you see here, and they're going to lower the activation energy. Okay? Okay, so once again, enzymes never change. They stay the same. And they are substrate specific. They have to find the correct sugar. Okay? Factors affecting enzymes to be denatured. So remember, denatured means it stops working. It's going to shut down. Okay, so that could be temperature, your pH level, substrate enzyme concentration, or coenzymes. Okay? And then we talked about just one model. Sorry, I should say one. Okay, the lock and key model. Remember, it has to bind to a substrate perfectly. So the enzyme has to bind to the substrate perfectly. Okay? And remember, the substrate breaks into products. Enzymes stay the same. Super important, this part right here. Okay, so make sure, oops, let me change this. So make sure you know this part. Okay, and just in case it's on your test, an inhibitor is going to stop everything from working. It's going to sit in the spot where the substrate needs to be, and it will no longer work. Okay, guys, so these are your notes over enzymes. I hope you have a good day. See you guys on